Let's jump right into today's explainer. We are doing a massive deep dive into Chapter 267, focusing entirely on genetic cardiomyopathies. It's an awesome opportunity to demystify this incredibly complex topic together. We're going to unpack the genetic architecture of heart muscle diseases, literally taking a journey from the microscopic machinery of a single heart cell all the way out to the expansive branches of a patient's family tree. It's just a fascinating look at how tiny biological blueprints have such massive clinical implications.
Okay, so here's our quick roadmap for today. We'll start with the genetic heart, head inside the myocyte, look at hypertrophic and restrictive phenotypes, then dilated and arrhythmogenic phenotypes, and finish up with clinical genetic screening. Let's get to it.
Section 1. The genetic heart, a silent inheritance. We generally think of heart disease as something we acquire, maybe through poor diet, lifestyle choices, or just getting older. But what's truly mind-blowing here is how often heart failure and sudden cardiac events trace straight back to silent hereditary factors hiding out in our DNA. Over 30%. When patients present with dilated cardiomyopathy, where the heart muscle is enlarged and weakened, and there are no other obvious causes like a bad viral infection or blocked arteries, over 30% of those cases actually show familial clustering. That is a massive proportion, and it perfectly illustrates why taking a detailed family history isn't just some clinical formality. It's a vital, life-saving diagnostic tool.
But before we go any deeper, let's get our definitions straight. Cardiomyopathy is strictly defined as a primary disease of the heart muscle itself. This is totally distinct from myocardial dysfunction that's caused by secondary issues, like ischemic heart disease from plumbing issues in the arteries or faulty leaking valves. We're talking about the actual muscle fibers being fundamentally flawed right from the start. And that sets the perfect stage for our genetic exploration.
Moving on to Section 2. Inside the myocyte, the cellular blueprint.
Now, we're going to zoom way in on a single heart cell, the myocyte, to figure out exactly where these genetic variants strike. This diagram serves as our master blueprint of the cell, mapping out exactly where structural proteins live and what they do. You can see these incredibly intricate networks of proteins that keep the cell structurally stable, allow it to physically contract, and connect it to its neighboring cells. When a genetic variant comes along and alters just one of these proteins, the entire system can completely falter.
So let's break this blueprint down into 3 major functional groups. First up, the sarcomeric proteins. Think of the sarcomere as the heart's engine. It's the actual contractile apparatus that does all the heavy lifting to pump your blood. Second, we have the dystrophin complexes. These act like the structural scaffolding of the cell, anchoring all that internal machinery to the outer membrane. And third, the desmosome complexes. You can essentially think of desmosomes as the cellular glue that binds adjacent heart cells together so they don't tear apart. A genetic mutation in any of these 3 distinct groups leads to vastly different types of cardiomyopathies.
Section 3. Hypertrophic and restrictive phenotypes, the thick and the stiff.
Zooming out slightly from our myocyte blueprint, let's contrast what happens to the whole heart when these specific systems break down. With hypertrophic cardiomyopathy, or HCM, the morphology shows a thick, overgrown muscle. You often see a highly thickened septum and a really small chamber. This is primarily driven by pathogenic sarcomeric variants, specifically in genes like MYH7 and MYBPC3, right there in the sarcomere engine. The physiology involves enhanced calcium sensitivity and severely impaired relaxation. Now, contrast that with restrictive cardiomyopathy, or RCM. This gives us a stiff, infiltrative heart. The chamber size might actually be normal, but the walls are severely restricted when it comes to filling with blood. This often points to genes like DES for Desmond or various metabolic storage diseases.
Jumping back to our cellular blueprint for just a second, look specifically at those sarcomeric proteins. Mutations in MYH7 and MYBPC3 account for about 80% of all hypertrophic cardiomyopathy cases. When this engine is built with faulty parts, it creates terrible, abnormal energetics. The heart desperately tries to compensate by overgrowing, which leads to massive cellular disarray. The muscle fibers literally become misaligned and tangled up in knots. And at the microscopic level, we can clearly see what causes that restrictive physiology we talked about. This electron micrograph shows Fabry's disease, which is a type of inherited storage disorder. Do you see all those concentric lamellar inclusions? Those are abnormal metabolic products physically cramping the cell. Because the cell can't clear out these glycosphingolipids due to a missing enzyme, the heart muscle becomes incredibly stiff and restricted, leading straight to RCM.
Section 4. Dilated and arrhythmogenic phenotypes, the thin and the broken. Now let's see how all this builds into two distinctly different phenotypes, where the heart muscle either heavily stretches out or just completely disconnect. Looking at this gross specimen, wow, it is truly striking. You are seeing the macro-level damage of dilated cardiomyopathy, or DCM. Look at that massive left ventricular dilation. The chamber is hugely enlarged and the walls appear stretched and totally thinned out, even though the heart's overall weight has actually increased. A heart like this simply cannot pump efficiently. The mechanical stretch causes the ventricle to completely fail.
If we zoom back in to the microscopic level for DCM, we can see what's actually happening inside that stretched out tissue. Notice the pink interstitial fibrosis, which is essentially just scarring, along with myocyte hypertrophy. Look at how the cells themselves are enlarged with these irregular dark nuclei. These destructive changes on the cellular level are exactly what drive that massive chamber dilation we just saw. So what's causing this one? Back to our trusty myocyte blueprint. I really want to draw your attention to a protein called titin, labeled here as TTN. Titin is this giant molecular spring that literally helps the muscle snap back after stretching. Truncating variants, meaning the genetic code gets abruptly cut short in this giant spring, are the most common cause of dilated cardiomyopathy. They actually account for up to 25% of familial DCM cases. If your molecular spring is broken, the heart stretches out and it just cannot spring back. And that brings us to our fourth phenotype, arrhythmogenic right ventricular cardiomyopathy, or ARVC. This is a brutal disorder where variants in desmosomal proteins cause the myocytes to physically disconnect from one another. The cells die off and get replaced by fat and fibrous scar tissue. This is incredibly dangerous because this fatty, scarred tissue is highly arrhythmogenic. It totally disrupts the heart's normal electrical pathways, creating a massive risk for sudden cardiac death, often in young, otherwise completely healthy individuals. Once again, the blueprint holds the answer. Look right here at the desmosome complexes. Remember how we called this the cellular glue? Well, when you have a mutation in genes like DSP or PKP2, which encode these exact proteins, the glue fails. Under the constant mechanical stress of a beating heart, especially during vigorous exercise, the cells literally pull apart, and that mechanical failure leads directly to the deadly electrical chaos of ARVC. Finally, Section 5, Clinical Genetic Screening, zooming out to the family tree. We've looked at the microscopic cells, we've looked at the whole heart, but now we need to look at the patient's entire family tree to understand why this matters so much in the clinic. When a patient presents with any of these phenotypes, the screening process follows a very logical progression. Step 1. Take a detailed 3-generation family history. You're hunting for sudden unexplained deaths, pacemakers placed at a really young age, or early heart failure. Step 2. Get a baseline echocardiogram and ECG to look for structural and electrical clues. Step 3. Proceed to specific genetic testing, guided by the proband, which is the first affected family member you identify. Pinpointing a specific variant in that one patient is incredibly powerful. It allows you to strategically screen relatives, potentially saving the lives of family members who had absolutely no idea they were carrying a silent threat. But there is a bit of a catch. Genetic cardiomyopathy features age-dependent and incomplete penetrance. So what does that actually mean? It means that having the genetic mutation doesn't mean you are born with a structurally abnormal heart. The disease develops and reveals itself over time, usually in adolescence or adulthood, and some carriers might literally never manifest the disease at all. And that is exactly why longitudinal screening, meaning checking in on these patients year after year, is absolutely critical. A perfectly clean echo at age 12 does not mean they are in the clear at age 25. Which brings us to a truly fascinating frontier. As viral vector gene therapies for Danon disease enter clinical trials, are we on the brink of curing genetic heart disease before it even starts? Danon disease is one of those restrictive storage disorders we talked about, caused by the LAMP2 gene. Scientists are now actively trying to deliver functional genes directly to the cardiomyocytes using viral vectors. It really makes you wonder, if we can fix the cellular blueprint at the source, could we one day erase these family inheritances entirely? It's an incredible thought to end on, and a testament to why understanding these genetic mechanisms is so vital. Your key takeaway here? Our genetics are a blueprint, for sure, but science is increasingly learning how to read, and maybe even edit, those plans. Thanks for joining me on this explainer, and keep questioning the science around you.